Welcome back to Miss B Biology. This is a revision lesson on the Osmosis required practical. Students, grab your book and grab your pen and also grab a past exam question on Osmosis required practical. I want you to complete the question after you have gone through this video and let me know in the comment section if you got full marks. You should. The aim of this practical is to investigate a range of concentrations of sugar solution on the mass of plant tissue. Students, it could be salt solution, right? And for plant tissue, it could be potato. That's what most students use in their practical at school. But it also could be carrots or any other type of plant tissue, all right? But the answers will always be the same. Let's move on. Step 1. Use a cork borer to cut out 5 potato cylinders. Step 2. Use a scalpel and a ruler to cut each potato cylinder so that they are the same length. We will be using 3 centimeters for each potato. This is our first control variable, having all potato cylinders at the same length of 3 centimeters. Remember, a control variable would be the variable that is kept constant, the variable that is kept the same. Next step is to measure the mass of each potato and record your result in a table. So this is our results table. So far, we have added our starting mass or initial mass, and students, each of these will be placed in different concentration solution. All right, and then we'll fill in the rest of the table as we go through. All right, so let's move on. Step 4, measure 10 centimeters cube of each sugar solution and pour into each boiling tube. Label each tube with the concentration of the solution, just like what you're seeing right here. The concentration of the solution is the independent variable. Remember, students, the independent variable is the variable that the scientist changes, right? And you can see that there are different concentration of solution. Right here we have 0.25, 0.5, 0.75. So the concentration of the solution is the independent variable. Let us now look at the control variables for the solution. Remember, the control variable would be the variables that are kept the same or constant. So we have the volume of the solution. Remember, we used 10 centimeters cube of each solution. I'm underlining this word, volume. Students, make sure you use the word volume. Do not use the word amount. You will not get marks for saying that. All right, another control variable is the type of solution that is used. All of these boiling tubes contain sugar solution, and that is something that we have controlled. Step 5, add 1 potato cylinder to each boiling tube and leave for 2 hours. Step 6 is to remove the potatoes, blot them dry, and record the final mass. There's a common exam question that has to do with the drying of the potato. Why did the student dry each piece of potato before weighing it? The answer is because water on the potato surface would increase the mass. This is what our table is looking like now. So we have our initial mass or final mass and the change in mass. The change in mass is a simple subtraction, right? So we subtracted the initial mass from the final mass to get the change in mass. All right, let's move on. Next thing that we need to do is to calculate the percentage change in mass for each potato cylinder. And here is the equation that we're using. So we're subtracting the initial mass from the final mass, divide that by the initial mass, and then multiply by 100. All right, students, you'll not get this equation in the exam, so please ensure that you remember it. Here is an exam question, a common exam question. Why is it important to calculate the percentage change in mass? Please write this question down, and this is the answer. Because each potato cylinder had a different starting mass or different mass at the start. All right, students, let us use these two values to get some practice using the equation to calculate percentage change in mass. So the final mass is 4.70 and the initial mass is 4.20. So here is our equation. Let us now plug the values in. So we're subtracting 4.20 from 4.70 and then divide by the initial mass, which is 4.20, multiply by 100. Your answer is 11.9%. This is the complete results table now, students. All right, but let us look at a few things before we move on. If you notice these two values right here, they are positive. This means that the potato for these two solutions gain mass. All right, and then these three right here, they are negative. This means that the potato lose mass. And students, this is where the exam questions will come in. Why did these two potato cylinder gain mass? And what caused these to lose mass? Right, and that's where even your definition for osmosis will become important. You need to know your definition because your definition can give you the answer to these exam questions. Okay? The last thing that we need to do is to plot, draw, and interpret our graph. Remember, the graph is change in mass against concentration of sugar solution. Remember, the independent variable, which is the concentration of sugar solution, must be on the x-axis. All right, let us draw this graph. Students, please do not judge me. Make sure yours is very neat in your exam. But the tool that I'm using right now, I'm not so good at using it, so I'm trying my best. All right, not bad. So let us interpret this graph on the next slide. Let us now interpret this graph. So this point right here represents the potato that was placed in the distilled water. This is for the potato that was placed in the 0.25 moles dm cube solution. So everything above this line, students, all of these points right here, these are showing an increase in mass, and you need to know why the potatoes increased in mass. All the values, all the points below this line, students, these are showing decrease in mass, and you need to know why the potato decreased in mass. Here is a common exam question. What caused the change in mass of the potato placed in the 0.25 moles dm cube solution? They're talking about this one right here. Why did it change in mass? They didn't tell you what type of change it is. They didn't say that it increased or decreased in mass. And so in your answer, students, you must mention that. You must mention that the potato increased in mass because water moved from a higher concentration, which is in the solution, to a lower concentration, which is inside the cells of the potato across a partially permeable membrane by osmosis. Right, students, you must say all of this. This is usually a 4 marker. All right, and to say all of this, you'll get your 4 marks. Let us continue with the interpretation of this graph. All right, so the question, what caused the change in mass of the potato placed in the 1 mole dm cube solution? They're talking about this potato right here, and this is a decrease in mass. So what caused this potato to decrease in mass? All right, so here is the answer. The potato decreased in mass because water moved from a higher concentration, which is inside the cells of the potato, to lower concentration, which is in the solution, across a partially permeable membrane by osmosis. Right, so the exam question asks what caused the change. They didn't tell you what change it is, and so you need to mention the change. You need to say that this is a decrease in mass, and then you need to say why there is a decrease in mass, because water is moving out of the cells of the potato, because inside the cells would be higher concentration of water, and the water is moving to a lower concentration, which is the solution, and it is moving across the partially permeable membrane, and by what process? Osmosis. So this is basically the definition of osmosis, students. All right, last question. What is the concentration of the sugar solution inside the potato cell? Students, the answer is always the same. Wherever this graph line cuts the x-axis, that is where you need to read off to get your answer, right? So in this case, it is about 0.4. Students, the answer is wherever the potato is not gaining or losing mass, that is where you'll find your answer for the concentration of the potato solution, right? So on the 0 mark, where there is no increase in mass or decrease in mass, and that answer is 0.4 moles dm cube. Students, I hope that your understanding of this topic is way better. Remember to do the exam questions and comment and let me know how many marks you scored. And thank you so much for watching and all the best in your exams. Bye bye.